Hello, my name is Jordan Kennedy, and today I'll be talking about the Kunjin virus or Kunjin encephalitis. The Kunjin virus is part of the family Flaviviridae in the genus Flavivirus. It is also known as KUNV, Australian encephalitis, and Murray Valley encephalitis. It is from the same subfamily as the West Nile and Katunga virus, which is Japanese encephalitis, although all of these different viruses are antigenically distinct. The virus is also considered a zoonotic virus. This means that the virus can come from not just humans, but from other animals that act as reservoir hosts or symptomatic hosts. The disease itself is endemic to Oceania, which consists of the tropical islands of the Pacific, although there has been isolated cases in mosquitoes in Southeast Asia. The only human cases have been found in Australia and closely surrounding regions. It has been found all over Australia and is particularly prevalent in areas near wetlands and rivers, as would be expected with the mosquito-borne virus. The name Kunjin virus comes from a tribe which is located near Mitchell River in Australia. It is located close to where the first cases were reported. These first cases were isolated in southeastern Australia in 1917. This outbreak seen 114 people stricken with the virus and showing symptoms. Before the virus was identified as a virus, it was called Australian X disease to denote that it was unknown. In 1960, the first vector of the virus was identified from an outbreak that took place 9 years prior in 1951. This vector was identified to be Culex annularisosis. Outbreaks of this virus have occurred periodically throughout history. Following the initial outbreak of 1917, another occurred in the following year, 1918. This outbreak resulted in 67 cases that were isolated to the southeast of Australia. It was at this time that study on the Australian X virus began heavily. Following this outbreak, most cases were isolated to the north of the country. In 1925, a smaller outbreak of 10 cases occurred in the northern regions of Australia. The next large outbreak occurred in 1951, when 48 reported cases came up. There were 19 fatal cases, all of which were isolated in the northern regions. The study of this outbreak was what eventually led scientists to discover the vector for this virus. The last large outbreak occurred in 1974, with 58 cases and 13 fatalities. This was also isolated in the northern regions of the country. Since 1974, there have only been scattered cases throughout the country. From studies on the patterns of the outbreaks, it has been concluded that the virus has a natural endemic cycle which involves water birds as reservoir hosts. The Kunjin virus is medically significant because of its effects on human health and also human-related animal health, such as horses and cattle. Because of this, it has received much studying. The virus itself only results in symptoms in 1 of 500 infections. Most of these infections are asymptomatic. When symptomatic, the symptoms are mild to severe. Early infection results in sudden onset fever, anorexia, and headache. Late symptoms include brain dysfunction or encephalitis, lethargy or fatigue, irritability, drowsiness, confusion, convulsions, and fits. The most significant effect of this virus is its residual effects. These residual effects occur in non-treated or severe infections. In severe and non-treated cases that resulted in brain dysfunction or encephalitis, it is rare not to have residual mental or physical dysfunction. This can include memory impairment or partial to full paralysis. The Kunjin virus is typically not fatal, although its relative, the West Nile virus, is. The transmission of the virus is by the mosquito Culex annularisosis. The virus is passed to the human or other victim when the mosquito takes a blood meal. It is injected into the bloodstream and the virus spreads throughout the human or animal's body. The Kunjin virus has a relatively simple life cycle. It is passed amongst the reservoir host, which is water birds, by its vector, the mosquito. The reservoir hosts have the virus in their body but never express any of the symptoms typically brought on by the virus in a susceptible host. If a mosquito takes a blood meal from a water bird that has the virus, and then takes a blood meal from a human or susceptible animal, then the virus is passed while the mosquito injects its saliva while feeding. Because of this transmission method, the virus particulates are typically concentrated near the salivary glands of the mosquitoes. When the virus is within an appropriate host, it invades the membranous cells until it reaches the nervous system and the brain. Here it causes the symptoms described before. Diagnosis for the infection of the Kunjin virus is by blood screening for antibodies brought on by the virus. The test should be done in the acute or convalescence phases of the infection. A fourfold rise in the antibodies against the virus confirms a clinical diagnosis. Following a diagnosis, the infected individual should seek appropriate medical assistance for his symptoms. As with all viruses, there is no specific treatment for it. Instead, supportive treatment is used to help patients with an infection. This means that the patient is given treatment for each of the individual symptoms expressed. For example, if the patient exhibits respiratory distress, ventilation is used. There is no vaccine created for this virus due to its low infection rate among humans. Prevention and control of the Kunjin virus is the same for all mosquito-borne diseases, such as malaria and yellow fever. For personal protection against an infection, the use of insect repellent of appropriate strength, loose long-sleeve clothing of light colors, and avoidance of mosquito-prevalent areas at peak hours of activities is suggested. Also, for protection during nights, bed nets are very useful, as they prevent mosquitoes from coming in contact with victims. For regional protection, governments play an intricate role by controlling the available habitats present for reproduction. This includes removing standing water from areas where it could become stagnant. Thank you for taking the time to watch this video. If you're looking for more information on the Kunjin virus, you can visit the Department of Medical Entomology of the University of Sydney's webpage on the Murray Valley encephalitis and Kunjin virus. Also, you can check out the Center for Disease Control's multiple pages on the Kunjin virus. Thank you.